OK，谢谢大家来我们今天的 Journal Club。然后今天呢是我上，还有 Chloe 会present。然后 I'll be doing like the background, and Sean will be spotlighting like a specific research paper, and Chloe will talk about the industry. So yeah. 对，就是大家能看得到吧？OK，所以我上选的 paper 呢是关于 like genetic engineering CRISPR，然后是比较就是我感觉比较一个 holistic 的 background review would be in synthetic biology, because synthetic biology is essentially about like um editing part of the genome of like an organism, for example bacteria, which is what he is focusing on today, and to make that of human use. So yeah. 所以今天的这个 background 就给大家稍微讲一下 synthetic biology 吧。对。Um, so to begin with, Aletta 刚刚进来，应该把 Okay, to begin with, 我先讲个就是 what is synthetic biology. So synthetic biology, 像我刚刚说的 essentially involves like redesigning organisms for useful purposes by engineering them to have like new abilities. For example, they can produce like a substance such as like a medicine or fuel, or gain a new ability, for example, like sensing something in the environment. These are some common goals of synthetic bio projects. Some examples includes like in the past, people have edited microorganisms that harnessed for bioremediation to clean like 就是水 soil or air 里边的这个 pollutants, or even there are yeast that are engineering to produce like say rose oil as an eco-friendly and sustainable substitute for like real roses, that perfumers use to make luxury scents. 然后大家如果知道 iGEM 的话，就是就是这里有一个 iGEM. It's I'm not sure the 全称 of that, but iGEM 有很多类似的 synthetic bio 的 project，大家感兴趣的话，可以去看一下。然后他们 SBI Systems Bio Initiative 也有做很多相关的东西，对，因为我以前是在他们的 iGEM team，but anyways, but I guess like a common question is like synthetic biology 和 genome editing，他们这两个 concept 还是有一点区别的，because which both okay they both involve like changing an organism's like genetic code, but in synthetic bio, you typically stitch together long stretches of DNA and insert them into an organism's genome. And these synthesized pieces of DNA could be like genes that are found in other organisms, or they could be like entirely novel, like you just designed them. But in genome editing, you typically use tools to make smaller changes to the organism's own DNA. Yeah. 然后 at this slide, there are some like common like techniques, or technologies or organizations that are involved or like are crucial to the field of synthetic bio. 然后到后面也会具体提到几个。Yeah. So one of the questions have been like, oh, can you synthesize an organism's entire genome? So where is the limit to this whole field of like synthetic biology, right? So in 20, 2002, 2002, scientists in the United States have synthesized a viral genome for the first time. Viral genomes are much smaller compared to the genomes of most bacteria and microorganisms, but they have shown that it is possible to create, for example, poliovirus from scratch, and which kind of brought attention to the risk that synthetic bio could be used to develop like biological weapons. So there has been ongoing like discussion regarding like the ethics and morality of synthetic bio, but today we're not going to go into that. But yeah, there has also been a continued effort to like push the limits of existing like DNA synthesis technology. There's one project called Genome Project Write, or GP Write. They're seeking to like synthesize or write the whole genomes from human cell lines and the genomes of other like plants and animals important to like global agriculture, etc. And which is related to Human Genome Project, which is basically trying to sequence or read all the DNAs that make up the human genome. So there is ongoing effort, but we have not reached like what we are satisfied with yet. So I think this field is very exciting, 然后有很多的 potential. But yeah. So 现在想给大家讲两个就是 like the core techniques to synthetic biology. One of it is like genetic engineering, CRISPR. 我感觉大家对 CRISPR 都还是比较熟悉的了。This is actually a slide from this class called Molecular Bio by Alice Heiklin. 然后我觉得 Heiklin 讲这些 techniques 讲都非常好，所以我非常推荐大家去上这门课，叫 Molecular Bio. It's in the spring semester. But yeah, basically like I'm not going to go into that. I bet Sean will go into that a little bit. But it's basically CRISPR is bacteria's like from bacteria's adaptive immune systems, and we're now using it to like do genetic engineering. 然后就是可以 insert genetic codes into the organism's original genome. But yeah. And then another core technique is DNA sequencing. I bet intro bio 应该也有讲过, but this is like next generation sequencing, 我这个 slideshow 写的是. Basically what happened is you start with primers, 然后在 primer 的时候呢，你会extend 你的这个 single strand with these fluorescent like DD ATP or DD-TDP, these DD-NTPs, 然后当他们全部 chain terminate 了之后，他们就是可以根据就是他们有长短不同的，然后它可以根据就是 basically laser 去 shot 这些like DD-NTP 来判断是哪一个which one of the four it is， 然后 read out， 大家应该都看过这个 sequence analysis 的这个图，对 ，but yeah。And then I'm gonna go briefly to talk about like the timeline or development of the field of synthetic bio. But I think something noticeable is that in 2010, the landmark achievement is I think it's the first time where scientists completely synthesized a working bacterial genome. 因为我们先前说的是2002年，他们 synthesize 出来的是一个 viral genome， 然后2008年应该是 synthesize 出来一个完整的 bacterial genome, but it's not like working. But in 2010, it's the first like working bacterial genome. So it's like a landmark achievement that kind of showed that like DNA synthesis and DNA assembly could be scaled to like a megabase size, delivering some of like the biggest ambitions from the start of the century. And but just scaling this DNA construction would definitely not be enough, right, to deliver all the ambitions in the field. But yeah. 然后2001年 this is from like a review article from Nature, and then it's just summarize some of the most important like findings or like paper published in the years. 然后对，我觉得还有一个比较crucial 的是2012年的 DNA used for data storage. I think that's something very interesting, like data memory, right? That's like a heated topic these days. 然后2013年is the genomically recoded E. coli. And I think Sean G. paper would be based on E. coli, I think, right? E. coli 就是大家现在都I think 就是做 synthetic bio 比较常用的一个菌种吧 ，yeah， 然后这边有写到，14年 bacteria that sense and record in the gut, and E. coli again engineered with 6 bases of DNA, and synthesis of yeast chromosome, and and in 2016, I don't think it's written here, but Nielsen published this thing called Cello, which is a remarkable like end-to-end computer-aided design system for logic circuit constructions in E. coli. And I think that's a big thing, like this logic circuit. And I'm also going to give like 2 examples later. Yeah. 然后18年 CAR T cell with logic control. CAR T cell 不知道大家记不记得我上个暑假有讲过一篇 review paper on CAR T cells. So if people are interested in this technology, they can reach out to me and I can send you the paper. Yeah. And carbon fixation and engineered E. coli. I think these are some of the more like well-known projects these days. Yeah. And right now, I just want to give like a brief example of like a genetic circuits. I probably won't have time to go into both of them, so I'll just talk about the top panel, which is a memory circuit. So I think like in genetic engineering or like synthetic bio, these genetic circuits are a big thing. Where yeah, there's this. So basically, um, you can think of it as like a switch. For example, in this case, the memory circuit is like a switch. There's on and off, and there's two types of this memory circuit. One is like transcriptionally regulated at the top, and at the bottom part of part A, it's recombinase mediated. So in the transcriptionally regulated circuit, we can see that, for example, if there's like an environmentally responsive trigger, you can produce this molecule called CRO. It will like trigger the transcriptional production of this molecule called CRO. 然后这个 CRO 呢，它其实就是会去 inhibit 这个 gene 往左边走，就是说只有右边会开，就是说它相当于是 inhibit 了 the production of CL, or inhibiting the switch from turning into off. And the other way around, 如果 the circuit or not the circuit, like the DNA is producing CL, it will inhibit the gene to 往右边 to transcribe, or in other words, inhibiting the memory circuit to switch to on. So I just thought that's something interesting. And recombinase media is something similar, where it's like, oh, when there's like a trigger, you produce these recombinase that can do something to the DNA of the organism, for example, like DNA inversion or DNA excision, etc. But yeah. Yeah, and then last but not least, I'll just briefly go over like some of the future directions or like how we have been applying this. And I think Chloe 在她讲 industry 的时候应该会讲更多, but basically there's like a range of routes to target different areas of the body when you try to feed these bacteria into the body, including the gut, circulating immune cells, and solid tumors. There have been research or like clinical trials with this a specific strain of salmonella. I'm not exactly sure, but it can like target the gut mucosa, and then or like more commonly like the hypoxic tissue at the core of the tumors, but they have been engineered to release toxins. And there have also been like other strains that has been used to immunize circulating immune cells against common cancer antigens. And in the gut, there's also bacteria species that can be engineered to release like therapeutics directly within the gut, because the gut has this whole like microbiome that is constantly like interacting with the host to like either secrete certain metabolites to affect distant organs, or just more focused on the gut in general. So engineering like the gut microbiome has also been like a huge research field these days. And 我第一个 PI Harris 王台就是在做这个相关的东西。Yeah. So yeah, you can also try to like prime the immune system to tolerate autoantigens, and to protect your host from like microbial attacks and toxins in general. So yeah, I think that's all my presentation, but I would say there are a lot of interesting things in the field of synthetic biology or microbiology in general. So it's a very exciting field. Yeah. 然后 Sean, you can go ahead and start your presentation.
Just
另外，大家如果就是有问题的话，可以随时打断我们三个，然后问你的问题，对，就直接 unmute 就可以了。
能看到我的 screen 吗，各位
看得到，看得到。
？Okay. So I'm going to be talking about a paper that I'm
但
actually
是能
currently.
看见你的那个 notes
这个是我的
page，
notes 吗？
它不是你的 notes page，就是能看到你那个 notes 的 page。 So it's not full
Okay.
screen, just so you know.
Interesting. I've never done this. 这个呢？
嗯，这样好了，
这样好了是吧？好的，好的。嗯，稍等一下，我的屏幕上东西有点多，稍微 move around 一下。啊，现在可以了吗？可以
可以的，
看到了吗
可以的。
？OK，好的，好的。All right, so I'm going to be talking about a compact cascade Cas3 system for targeted genome engineering. This paper is authored by Balint Sorgo. I think that's how you pronounce his name. This is actually one of the papers that supports a lot of the theory for a project that I'm currently working on at the Harry Swan lab that Coco mentioned previously. So it's very relevant to my research, and I do think it's a very interesting and powerful genomic engineering system that has a lot of potential for therapeutic usage, which is actually what we're looking into right now. So I'll start off with just a general brief overview of what I'll be talking about. Um, Coco mentioned that CRISPR was one of the most widely used synthetic bio and genetic editing technologies available to us. And I think since as early as 2015, maybe 2018, it's become a sort of buzzword for genetic engineering. And then everyone, when they think about anything bio, CRISPR is one of those things that come up to them. So I would just like to go a little bit more into detail, specifically considering what CRISPR actually is. And then I will also go into the types of CRISPR enzymes there are and how they function very differently, actually. And why specifically we chose Cas3 as our sort of functioning vector in gene editing. And then obviously I'll discuss mostly the results from the Cascade Cas3 engineering that the paper discusses, just to show you guys, what are the benefits and what are the potential caveats of Cas3 engineering. And then there will be a discussion section at the end for some further implications. So as an introduction, CRISPR-Cas systems, when they were first discovered, are a diverse group of RNA-guided nucleases, so enzymes that break down nucleotides, that defend prokaryotes, bacteria, against viral invaders. So in their original form, they, they were essentially the immune system of bacteria. Um, but what we ended up seeing was that this can actually be leveraged against the bacteria themselves if we introduce targets that are tailored to our needs. So one of those problems that bacteria are currently causing is antibiotic resistance. In a lot of the hospitals and especially ICU intensive care units, where a lot of the strong antibiotics are used, what we're seeing is a lot of these bacteria are not responding. They are not sensitive to these antibiotics, and they just keep on dividing, and that poses a significant health risk to ICU patients. So the objective here that the author uh, aims for is to repurpose and optimize a type 1C, which I will go into further detail, for both endogenous and heterologous genome engineering in bacteria. And so what that really means is we're trying to draw or design our own target and insert that into bacteria such that they will eliminate things that we want to see gone, but using their own molecular machinery. So I'm just going to go through a few quick definitions for CRISPR-Cas systems. CRISPR stands for clustered regularly interspaced short palindromic repeats. I know that's a bit of a handful. They're really just DNA sequences in prokaryotes that contain short repetitions of base sequences. So what we end up seeing is that we have a lot of the hairpins, so sequences that repeat themselves, interspaced with viral DNA. So the non-hairpins that are in these CRISPR sequences, they are stored information from previously defeated viral the viral genome, so that when the bacteria matches whatever was stored in these CRISPR arrays with foreign DNA, it will immediately recruit either Cas3, 9, or 12. Those are the big three. To degrade that DNA to prevent a viral or phagocytic invasion, right? Cas are CRISPR-associated proteins. They are enzymes that use CRISPR sequences as a guide to identify and cut the specific strands of DNA. Be self-explanatory. And cascade is the whole complex. And that's what we see for Cas3 because it recruits more than one protein. It's a whole set of proteins that that come together to form this really complex antiviral defense line. And this is also what we're leveraging for the case of Cas3 in our genome engineering. So, like I said, I was going to go into a little bit more detail on the classifications of CRISPR systems. And this is really important because Cas9 is what we've sort of always heard that can do like really crazy things with introducing double-strand breaks and sort of creating these very versatile applications for how you can mutate a few base pairs in bacteria to create something completely different. Cas3 is different from Cas9 in the sense that as a type 1 multi-protein complex, so it takes multiple proteins to come together for Cas3 to work. It's more so a helicase nuclease. So what it does is it sort of gets rid of the twisted double-stranded DNA structure. It irons out that twist, and then it starts degrading DNA processively, which means it doesn't just cut it in the middle. It sort of like sets itself on the DNA and just starts eating it both ways. So it's bidirectional degradation. So Cas3 in this sense is much better for large scale deletions, which is really critical to our applications. And is what really sets it apart from Cas9, which has been studied relatively more thoroughly. And so as you can see here on this picture, Cas3 degrades DNA targets identified by a separate complex. And this complex and the guide RNAs associated with it is what we'll engineer. So the problem with Cas3, right? Like it's not like this thing exists in bacteria and it's just ready to go and work for humans already. The main problem with Cas3 is that it really is limited to its native hosts. So if say P. aeruginosa has Cas3, it hasn't really been applied to other bacteria, which really limits is like area of functionality. Um, and then the Cas3 complexes are also really different for different bacteria. So every different type of bacteria has a different Cas3. That's what makes it really hard to engineer and have like a Cas3 system that applies to all different types of bacteria. So P. aeruginosa is one of those bacteria that really sort of caught the researchers' attention because it's streamlined and only has 4 proteins. It's counter selection free, meaning that you don't have to, apply antibiotic. You don't apply like antibiotic resistant, like an AMP selection on the plate to make sure that Cas3 works. It's just sort of there already able to be used. And it's also programmable, and we talked about the scale issue, so it's really ideal for what we're going to do here. And I'm just going to briefly go over the results here. So Cas5, 8, and 7, those are your complexes, one of them at least, that produces the CRRNA, which is CRISPR RNA, which is what we'll engineer. The CRRNA guided surveillance watchdog. So that complex is what searches actively for foreign RNA. And once it identifies that foreign RNA, it recruits Cas3, and Cas3 starts the degradation process. So a few observations that the researchers noticed during their experiments. Introducing genome targeting plasmid when induced, when you turn the plasmid on and transcribes the information, well, transcribes the information on the plasmid, it actually causes a reversible growth delay. And this has been observed in many other experiments, and we don't exactly know why. I will move on to the other stuff. Um, the researchers were able to target this one protein called PHCM. It's like a pigmentation protein, and so if you target this and delete everything here, you're gonna see yellow bacteria. You're gonna see the whole media look yellow instead of green. And then there's also a lot of PCR work done based on sort of providing evidence for the bidirectional degradation from WGS, which is whole genome sequencing data. Um, and yeah, so there are a few pictograms here that I would like to talk about. RSR on picture A really just means the repeats and the sequence that of interest, which is what we are going to engineer. Uh, the S thing here is really like the selectable target, right, that we can engineer. And then once we sort of select the target, Cas3 is going to make a complex out of all these things and just start degrading DNA like this. And this is the growth delay that I was talking about from the OD600. It's OD stands for optical density. And so it really is just like a sort of general indication of how much bacteria there is growing at any given point of time. And so we see that for non-targeted bacteria, growth is much more immediate than their targeted uh, counterparts. And we don't exactly know why, but whenever you target these proteins, you'll see that the bacteria just takes its time before it really wants to. And then over here, this is a graph that really shows how the whole genome sequencing data shows that the protein degradation is bidirectional because your target is in the middle here, and then when they sequence stuff, when they sequenced everything, so the whole genome, they realized that the dotted lines here are parts of the PCR that yielded no product. So you can see that both sides of the genome have been degraded.
想问
Uh,
一个问题，
yeah.
就是你上一张图那个图 C，它不是有 minus inducer and plus inducer and plus inducer
Oh yeah.
recovery? What's recovery? Just wondering.
Recovery 就是I'll come back to that, but I'm sure it
Okay,
means
okay,
something.
yeah, yeah,
Yeah,
yeah.
yeah, yeah. I'm sure it means something. I'll come back to
Okay.
that. Yeah, so just a little bit more on the graphs. I mentioned there was a debugging session sort of in the paper where they talked about how spacer excision is a problem with the CRISPR RNAs. So what they ended up seeing is that the guide RNAs that we try to engineer to target different proteins, they like to loop against themselves. And when that happens, and when that makes a hairpin, it actually loops out the functional part, the part that we designed. And so when that happens, we have no deletion, right? And so the solution that the researchers came up with is to modify the plasmid, especially the areas flanking the CRISPR RNAs, so that as you can see here, so that they don't form these hairpins that are going to eliminate transcription from the programmed sequences on the plasmid. And so as you can see over here, the editing efficiency of the modified repeat, which is where I mentioned that they edited the sequence to eliminate excisions, they were much better than our wild type repeats, which is when they didn't have this extra bit of engineering going into their plasmids. And GraphC really just shows that somehow, like even with their re-engineered plasmid to deliver the CRRNAs, the bacteria still takes its time before it wants to go, and we don't really know why. And so I'm going to talk about D, E, and F over here in a comparison that the researchers did between Cas3 and Cas9. This is really just to highlight the fact that Cas3 is much better at making large deletions and making accurate deletions at that versus Cas9. So Cas9 is so this really just shows that Cas9 is more so geared towards making point mutations. It's good at like cutting 1 to 2 base pairs versus a thousand or even more. And so these graphs over here, like for 170 kilobases, 500, and then 249,000, Cas3 has much better efficiency because of the suspected recruitment of homology-directed repairs, which is just that the bacteria uses external templates to fix up all the deletions made. And then here you can see that Cas3 is much better at making a lot of edits, a large deletion versus Cas9. And so some of the practical targets that this Cas3 technology can be used is E. coli. I'm just gonna skip through this. They did an experiment on lactazine, and there was like somewhat high survivability there, and also Klebsiella pneumoniae, which is what I'm working on currently. So this is one of those really like persistent bacteria in ICUs, and they were basically able to show evidence that the cleaving and the Cas3 effects apply to this type of bacteria as well. So what are our takeaways, right? High efficiency and large deletions because Cas3 is just able to delete 424 kilobases with a mean and median of this and that, pretty high compared to Cas9. It's bidirectional, talked about that. It's versatile because it's worked in P. aeruginosa, E. coli, P. syringae, and K. pneumoniae. And HDR efficiency, meaning that its recombination effect is good, and it can repair the double-stranded breaks, unlike Cas9. So I'm just gonna quickly mention the research that I'm doing now. The hope is that Cas3 will work via plasmid engineering. I'll deliver it to K. pneumoniae, and then within K. pneumoniae, we're going to eliminate two things. One is genomic DNA, so part of the bacteria's genome that has resistance to standard of care antibiotics, and the other thing is plasmids within K. pneumoniae that has resistance, which we are going to eliminate through Cas3. And the vehicle that is going to deliver Cas3, this is the second layer, is TP114, which is really far down the line, but that is sort of the hope of what we're trying to do here. And so, yeah, that's all. Thank you, everyone, for listening. And if there are any questions, I'll be happy to answer.
大家都有什么关于这个 paper 的问题，可以现在提出来。然后如果有更 general 的问题，也可以等 Chloe 讲完再问都行。那么我们要不要先 Chloe 你先开始你的 presentation？
好的，没有问题。那我 share 一下我的屏幕吧，我看一下。我看到我们讲的是 microbiome，所以我可能跟其他两位有点偏题。我就是看的是那个
没
小
关系没关系，
的
差不
market。
多都可以。
好
Yeah.
的，好的，早知道我看 sequencing 了，但是没有问题，我先 share 一下。
No, it's okay. 没问题没问题。和 bacteria 相关的都可
好，可以看到吗？可以看到我的 share screen 吗？还是还没上去？
以。Yes，看得见看得见。
看得见，OK，那我把它 zoom in 一下。好，那我今天就讲 microbiome 的 industry 吧。然后我找的是一家比较小的，偏初创公司，然后他们自己是 developed 了，2023年刚拿到一个 FDA approval for 他们自己的 medication。然后我就一点点讲吧。所以我先 fill in 一下这个 gap。这个 microbiome 呢，我们现在的 focus 就是collection of all like including like bacteria, fungi, viruses and their genes. And so like a lot of that is understanding how they kind of function and balance within our bodies. And so 我们今天先看一下这个 microbiome 的 market overview。好，它其实是个 relatively 的 niche market，因为其实很多 major players 都是那种初创公司。然后呢，它现在的 microbiome market，in terms of revenue，其实只有0.3个billion in 2023。 但是呢，it's projected to become 1.7 billion in 2029. So 我们可以看到计算过后呢，这个 compound annual growth rate 其实是 36.1%，所以它是有很大的增长空间的。由于它是一个比较 niche 的一个小 sector of the overall pharmaceutical market。然后这几个 recent progression trend 呢，其实是有三个重点。一个呢就是近几年大家由于生活水准、教育水准慢慢往上走，然后有一个 increasing awareness of microbiome's importance on the human health and pharmaceutical sector。然后呢，还有一个就是随着可能大家的就是 income 越来越高，然后大家的对于这个 health 的 demand 越来越高，有一个 increased demand for personalized medicine，所以不再是 generalized medicine，而是根据我自己身体里的这么一个 pH 这个 overall balance 跟我身体里的一些 bacteria 来给我调整一个我的 personalized medicine。然后第三点呢，就是前面二位也说到这个 progression of sequencing technologies 有去帮助这个市场去 grow，然后去 develop。好，那我今天要讲的就是这个 Seres Therapeutics，它自己给自己的介绍就是一个 commercial stage microbiome therapeutics company，然后它是 focus on commercializing biological drugs that restore health by returning the function of disrupted microbiome to a non-disease state。然后这个公司现在已经有14年了，它是 founded in 2010，然后 based in Massachusetts。然后2023 was a big year for Sears because they acquired FDA approval for their Voust, yeah, which is this orally administered microbiota based therapeutic indicated to prevent C. diff for adults. And they were also named the 100 top innovative companies of the year in 2023. And the main reason I picked this because of all of the small microbiome companies I was looking at, this one was the first I came across that actually had an IPO, which meant that their financial data was pretty public and was easy for me to access. So yeah. And then so let me just address the pain point that they're trying to address right here. So what is C. diff? It's basically this bacterium that resides naturally in our guts, right? It doesn't do much harm until there's this disruption in the balance of the gut microbiota. And a lot of this disruption actually occurs from an consumption of antibiotics, which can kill a lot of harmful as well as beneficial bacteria inside our guts. And so a leading result is diarrhea and inflammation of the colon. And it's actually quite a widespread problem because the CDC estimates that there are 500,000 C. diff infections across the US in every year, and some might even be life-threatening. So if we look at the Sears company finances, we can see that it's plummeted quite a bit over the years. At its initial IPO in 2015, it had a stock price, I believe, of $18. It has now fallen to $0.99. It has a current market cap of 151 millions. So basically, market cap is you take the stock price and you multiply it by the stock quantity and you have the market cap. And then it has a gross revenue of 126.32 million. And while it has fallen a bit, I personally, after analyzing this company, am optimistic that it will grow because 2023 was a great year, and I hope that it will turn this decline around. So basically, after all of its decline, it built its own program, launched at restructuring its financial and administrative organization. It has plans to cut down costs, and it has plans to achieve and increase sales in the workforce and across its market. And then it also partnered up with the Nestle Health Science in 2023 to launch its new product. So if we look at the competition, there's actually quite a bit because a lot of other companies have they have more resources, they may have more big name collaborations, and there's also the possibility of M&A, which basically create synergy and even more researchers and R&D and investments for them. And I guess with this particular market, a lot of it is focused on what they can do. It's more product-based than market-based. A lot of it relies on technology and R&D. So for this particular market, I think there's a lot of competition when it comes to investors, and there's a lot of competitions when it comes to who has the most efficient product on this market. And some of them are like CryptoPort, Ultimune, like Siga Technologies, like things that are quite out there and are already developing their new and own products. So I guess out of all of the market competition and growth, one thing that I noticed is that an advantage of Sears is that they have a price target of $5, and many investors have estimated that it has a potential upside of 400%. So that is one of the good things in this market when even facing all of these competitions. Like this list is like 4 lines long, so it's quite a bit of competition. But if we look at its first quarter financial report status update of 2024, they're hoping to kind of ground their market with the valves that they have developed. This I guess for them is their market advantage because it's the only and it's the first and only FDA approved oral administration of this type of medication that addresses C. diff. And a lot of their progression focuses on getting more patients enrolled on this program, getting more patient data, and then increasing their sales and manufacturing in 2024. However, this company currently functions at a net loss of 14.1 million in the first quarter of 2024, but it has decreased from a net loss of 71.2 million for the same period in 2023. So across 1 year, they've made up for the 30 million loss. And they're looking also to kind of reduce R&D expenses for the first quarter because it is the majority of where the costs have gone to. So a little bit more about this company's specific approach to medicine is that they use something called the Microbiome Therapeutics Engine to design, test, and manage the multifunctional bacterial consortia. So it's a 4-step process where they first gather insights on existing clinical trials and studies, and they kind of undergo their own mechanisms of analysis into selecting these target biomarkers to market these target microbiomes, and that's when they kind of identify and they kind of navigate the entire disease pathways. And then after that, they kind of try to develop their own consortia and then test it with in vitro assays and then to test these properties. And then number 4 is that if these work and if the test results come back favorable, they undergo a cycle of maintaining governmental regulations and approvals, and they speed forward to production, which is what they did with the VOST, which that they're currently developing. Yeah. Okay. And then so I guess from this company, we learned that a majority of these kind of niche biotech companies, a lot of it comes from the technology that they have, a lot of it comes from the database that they have access to, and their patient market size, and which directly correlates with the amount of data and the quality of data that they can get their hands on into developing a overall functioning medication. And so I wanted to show you guys the website that they built. If I can just, yes. So as you can see, they have a functioning medication that is FDA approved.
我们好像看不见 website, Chloe.
哦，你看不见吗？哎，等一下，
对，
我切下屏。哎，我怎么切屏？算我把它拉过来吧。能不能拿过去？看到了不？
还是看不见，你可能要 unshare 然后重新 share 一下。
好的，我想想啊。 怎么
找
unshare 来着？等一下。哦，OK，哦，我看到了。OK， can you?
Yeah.
Yes. Okay. So basically, this is the website I wanted to show you. This is the kind of the treatment that they have spent the past 2 years developing and acquiring an approval for. And so it was approved in 2023 and it started kind of mass producing in 2023, and 2024 is when they expect to pick up the pace in manufacturing and sales. And yes, that is all I have for you today. Thank you.
谢谢 Chloe，我先 stop recording。